So we went through koala SNP analysis. We went through the Human Genome Project. You can talk about that. But for this question, it's conservation management. Micah, what example are you going to use in your exams?
Koalas.
Yeah, very good. So you can talk about koala haplotype analysis to look at genetic diversity. And that simply allows us to allocate our time and energy towards koala subpopulations that have the lowest diversity that we need to manage with the highest priority. Makes quite logical sense. Abinan, what is a haplotype?
Doesn't it like measure the amount of like SNPs, I guess? Like a different
Collection of SNPs. That's all it is.
Yeah.
Collection of SNPs. A group of SNPs is one haplotype. Very good. All right. Question 20. Those of you who haven't seen it, you have 30 to 40 seconds to have a look at it before I start picking on you. I don't need an answer. 40 seconds, you better have a game plan. Okay. What is this question actually testing, everyone? So Maya, you see this question in your exam. What am I testing here?
Mitosis? Wait.
Oh.
Meiosis?
Yeah, meiosis. Right. But more specific, what am I testing here? Micah.
Like how different alleles combine.
Still a bit vague. Raiden, what am I testing here?
Oh, your knowledge of random segregation.
Yes, that's what I want to hear, right? Because if you don't know what the question's testing, you don't know what to say to get the most marks, and you don't know how to plan your answer. So right now, your head in this huge expanse of HSC biology should narrow down into thinking about meiosis and should narrow down to thinking about random segregation and independent assortment. The way I know this and the way Raiden mentioned it is because it's saying assume that crossing over does not occur. Now, if crossing over did occur, what can happen is any of these alleles on homologous chromosomes can interchange. But now we know that can't happen, right? And this is where you have little thought experiments in your head, right? There's going to be 4 gametes formed in this process, right? What's going to happen within a homologous pair? Bilal, within a homologous pair, what process occurs?
Mitosis?
No. Within a homologous pair, mitosis, we're talking about random segregation and
random segregation. It will be random segregation.
Yes, right. Remember, mitosis is a completely different process, right? You're on a different page then. So we're talking about random segregation here. So if this chromosome goes to the left, this must goes to the right. So they must go to opposite ends. And the same applies here. This chromosome can go to the right, has a 50 chance of going in that respect, left or right. And this chromosome has an equally likely chance. Now, what this means in letter sense is that all we need to have is a combination of one of these chromosomes and one of these chromosomes. That will make up the organism's genotype. We all agree, right? Because the organism is inheriting one big chromosome, one small chromosome. Okay, so let's go through this. What do we have here? So let's start with option A. Sean, what do you think about option A? AB little c. Can an organism inherit AB little c?
No.
So apologies, I didn't clarify. Capital A, capital B, little c. Can it inherit that?
Oh, wait.
Do you see a chromosome which has capital A, capital B, little c?
Yes.
Yes.
Okay, good, right? So the answer is yes. What about here? Yes or no? Maya, your turn. How about the D and E? What do you think about that, Maya? So you see a capital D and capital E. Can we inherit a capital D with a capital E?
No.
No, because if you have a capital D, you must have been inheriting this chromosome, and unless crossing over occurred, a capital E could not have jumped across. Do we all understand that? Right? This is actually quite a simple question. So A is out of the question. Have a look at B. Can someone tell me why B might not be right or why it might be right? A couple seconds to have a look at it. Go through the same thought process. Since crossing over did not occur, this organism is going to get one large chromosome, one small chromosome. You can't violate that rule. And if these combinations work out, Raiden, why did they not work out?
Because for the first one, a lowercase b can't jump over to the first one.
Yeah, you see capital A, little b, and a capital C. Capital A, little b makes sense. It's on the same chromosome. yeah, capital A and little b, but you see a capital C, that makes sense. So capital A, capital B, capital C doesn't make sense because that capital C must have broken off, right? And we've clearly said crossing over does not occur. So B is not right. Alec, what do you think about C? Go through the same thought process. See, this question, a kindy student can answer it. What the hard part about this question was understanding what the question was asking. And all you need to realize is you can only pick one big chromosome, one small chromosome. You can pick any combination you'd like. So Alec, what do you think about option C? Does this work out? Capital A, little b, little c. Capital A, Alec, little b. Can you have a little c, Alec?
I'm pretty sure his audio doesn't work.
Oh no. Okay. Who can I ask? Bilal. Back
No,
to
it
you.
doesn't work.
No, it doesn't, right? So we cancelled all three out. Test this one, everyone. Does that work out?
I think it does.
It does.
Yeah, it does.
Tick. That's your answer. Not too hard, right? Good. Did I clarify that for everyone? Did it make sense?
Yeah.
Right? If crossing over happened, then quite literally any allele combination can occur based off this. Right? So I guess one question you have to realize, a long response version of this question that they love to ask is contrast the genotype possibilities. It'd be a 6-mark long response, right? Contrast the genotype possibilities if A, crossing over did occur, and B, crossing over didn't occur. 3 marks. So maybe what you can do is you can quickly screenshot this and think about it for a long response point of view, right? I want you to just have a plan in your head. If you can even think through a plan in your head, I'm happy that you understand this process going on here.
Wait, so what's the actual question that's the long response is asking?
Contrast the genotype possibilities, or it'll
Okay.
be a broader question because contrast specifically means differences. It'll be critically
You
compare.
could say discuss.
Yeah, you could say discuss. Critically compare the genotype possibilities if crossing over did occur and crossing over did not occur. 6 marks. Give it a go. If you'd like to write a full written answer, do so and send it to me and I'll give you a verbal feedback and a mark, right? But I'm just showing you the different possibilities this question can go to. It's not that it will be that difficult, but good. All right, happy to move on. Good. I'm happy that, good job. Those of you picked up what this question was asking, that's what I'm looking for here, because then you know how to approach the question. All right. Sean, this question, is this from the additional question booklet? It looks
This
like
is from last week, so I'm not completely sure.
all right. Okay, let's go through it. Okay. Flowchart illustrates the effect of a point mutation. Abinine, missense. Do you mind clarifying what mutation that was? Because I asked you that last lesson, so I want to get the recap answer. What is a missense mutation?
So when you get the codon changing, I guess, due to the base substitution, or it's a stop codon, one of them.
0 out of 1 so far. Maya, your turn. What is a missense mutation?
A change in a substitution in a single base that results in a change in one amino acid.
Good, good. So the key thing here is when I ask you all questions in this lesson, you need to give me exam answers back. I'm going to quite literally mark you on the spot based off your verbal answer. Because if you can't verbally say it, you can't write it in an exam. There's no hope there, right? So when you think about a mutation, you should all by now be able to say to me that it could be at a chromosomal level or at a base level. And a base level is a point mutation, right? And with a point mutation, you can have a substitution or you can have a frame shift, which is your addition or deletion. If you have a substitution mutation, Micah, what are the 3 types you can have again?
Missense, nonsense, or silent.
Good. And missense, what's the key buzzword you want to have in a definition?
Like it's a change in one amino acid.
Very good. That's what I was looking for, Abhinay, in your answer. It's a point mutation, a point substitution mutation that results in a change of one amino acid. Not stop codon, because what is a stop codon mutation, Miriam?
A stop codon mutation is a nonsense mutation.
Yeah, very good. So nonsense mutation produces a stop codon. Good. I mean, do you just mind muting yourself? Only because there's a background frequency that I can hear. Yeah, thank you. You feel free to unmute yourself, everyone, when I do ask you a question, though. Good. So nonsense mutation produces a stop codon, and silent, produces the same amino acid. Good. Now, what you see here is a point mutation, and what has occurred? You've gone from TCT to where? ACT. Do you all agree? That's the effect here. It's a base substitution. What's happened here? What is this representing? Number 3, Sean?
Is that meant to be the RNA to it?
mRNA. You need to be
mRNA, sorry.
very
My bad.
specific. Okay, remember, 2 weeks out from the HSC, you have to be very specific. Bio is not hard. It's just an exam of articulation and specificity. So focus on the detail there. Good, that is the mRNA strain. You see uracil, so straight away you think mRNA. So what do you think you should be representing here? Because I stressed this to you. With a long response about mutations, you need to link 3 things. What are the 3 things you need to link? If they say this mutation has happened, how's it going to affect the organism? What are the 3 things you talk about?
How it affects the base sequence, and
Good.
then the polypeptide, and then the protein.
Yeah, so the way you should be thinking of it is, how does it change the DNA? So what kind of mutation is it? So identify that, right? And then you have to explain the effect on the RNA, then the effect on the amino acid sequence, and then you link it to the polypeptide and the phenotype. Does that make sense? So that's the structure I have. Make sure you engrail that into you remember that, okay? Because it's very important. Good. All right. So outline the series of events from stages 2 to 4 that resulted in the faulty protein. So here we know we are talking about the amino acid sequence, right? What did I tell you? Look at the DNA, then you must look at the RNA, then you must look at the amino acid sequence. So series of events straight away. Let's see. Ishan, what are they actually testing you here?
Like what they're questioning us on.
I'm looking for 2 words. You either know it or you don't know it. What are the 2 things?
Transcription, translation.
Very good, right? That's all you need to realize. So the question is quite literally outline transcription and translation, but refer to the letters you see up here, right? So Ishan, now's your chance to explain to us, the class. Can you describe transcription to us? Exam answer. I don't want to, ifs and buts or some things and things. What happens in transcription? Clear, concise answer. Go.
In transcription, mRNA template is formed, which can I don't know how to word it.
Okay, let me ask Braden.
Can I go?
Yeah, Zachariah, give it a go. Sorry, I couldn't see your picture, so I didn't ask you too much. Good.
That's all right. The process or the purpose of transcription is to create a template strand of DNA that is able to float into the cytoplasm and reach the organelle. Damn it, how did I forget this? The ribosome, where it is translated in the next stage. For transcription, the template is created via damn it.
That's okay. That's okay. This is why I'm putting it out. So you all realize that you all know this stuff, but you all need to work on your articulation. Right now, your articulation, for all of you, is not at a full band 6 range yet. So you need to focus on fine-tuning that. Maya, your turn. I want 1 sentence and I want the keywords. Go.
in transcription, mRNA is a complementary mRNA strand is created from the DNA template using RNA polymerase.
Perfect. She's mentioned complementary. She's mentioned RNA polymerase. It's the only other thing I'd mention is that uracil is replaced instead of thymine. If you mention these three things, right, you've got the crux of transcription. Remember, this is a 3-mark question. If it was 5 marks, for example, then you start talking about 5' guanosine cap and polyadenine tail, poly-A tail, all of that stuff, and splicing out your introns. All of that is extra. You don't need it. And this is why you need to do exam questions, right? Students know all this stuff, but the poor students will just verbal garbage everything they remember. Good student would know what are the key things I'm looking for in an exam. If you don't say the word complementary, you're not getting the mark. If you don't refer to the letters here, you are not getting the mark, right? So a template exam answer would look like this, right? In step 2 to step 3, the process of transcription occurs, whereby the template GATACT strand is converted into a complementary mRNA molecule with thymine replaced by uracil. The enzyme responsible for this is RNA polymerase, full stop. You see how I referred to the data? I've used the buzzwords, and it's clear and concise. Everyone with me, right? Now, I don't want to waste too much time. You guys should be a bit faster with this. I'm going to pick one person to do better and explain translation well. So how about, Miriam, throwing it to you now. Translation, what would you like to mention?
The mRNA strand travels to the ribosome, which and then it is formed into amino acids, which bind into groups of 3, creating codons, which then bind, creating a polypeptide.
Good. But why did you go from amino acids back to codons? Because codons are still the language of mRNA. And the second you say amino acid, the mRNA is coded for the amino acid. You don't need to mention codons. The mRNA is structured as codons. Does that make sense? So being specific here, you need to mention mRNA codons will affect the tRNA anticodons and the amino acid sequence. And one thing you missed is a tRNA that I'd like you to mention, right? One more person. Let me ask Raiden. Give it a go. Translation.
In translation, so mRNA travels to the ribosome where it's decoded outside the nucleus and it produces a amino acid chain.
Okay. Now, with this question, is there anything you'd like to mention, Raiden? Specifically, because you gave a generic description that could answer any.
You have to mention the mRNA to the amino acid from step 3 to 4.
Yeah, good. Any data, any other data we'd like to mention, everyone, to be safe? To show that we're answering this question and not a general.
UGA. The 3' to 5' direction?
No. That's going way outside this question. There is no need to mention 3' or 5'. Do not mention that unless it is given in the question. Okay, that's a big learning point. Very low yield. Come on, guys. How many codons do you see here? You mentioned the word codon in all of your answers, but why is no one talking about the number of codons? Yeah, so how many amino acids would this produce?
2 amino acids.
Good. And why would the protein be faulty, everyone? What does it mean to be a faulty protein from a substitution mutation? 2 mutations could have happened to make it faulty. What are the 2 mutations? Abinan, your turn. What 2 mutations from substitution can turn the protein faulty? I gave you 3 here. Kind of.
Is it missense and missense and the one which changes the stop codon?
Yeah,
stop
I don't.
nonsense.
Yes, nonsense. Very good. I don't want to hear this one or that one. I want specific answers. The missense or nonsense mutation, right? So to be very specific, yes?
I was going to ask, with the nonsense mutation, since it's non-functional, would that be a faulty protein or is it not counted?
A nonsense would be a very faulty protein because I guess the way you think about this is not the entire amino acid sequence, the TCT, right? It's much longer than that. So imagine there's another 500 amino acids in the normal polypeptide. If you cause a nonsense mutation and you produce a stop codon, which quite literally just codes for release factor. So instead of an amino acid binding, release factor will bind, and everything to the left of that is going to get released, aka that's going to be the final polypeptide. You're not going to produce whatever 500, if this was 500 bases, whatever 500 divided by 3 is, you're not going to produce those many amino acids, right? So you're going to have a severely deficient in size polypeptide chain. Does that make sense? Nonsense is the
Yep.
worst. It's going to make a worse faulty protein than a missense mutation, right? So what I would mention, the key things I'd mention here is translation is a process whereby the ribosome will move along the mRNA codons, attaching complementary tRNA anticodons in a specific sequence of amino acids, full stop. Do you see how I've spoken about codons, tRNA, amino acids?
Yep.
Yes?
I was
That's
just
it.
a bit confused because our teacher told us that if a mutation is generally deleterious, then you could say the protein is faulty. Otherwise, it's not. And I wasn't sure with like nonsense
The
because
mutation is
the protein
deleterious.
isn't even made.
Deleterious just means bad in broad terms. It's just a fancy way of saying bad, right? And so it can be bad through a nonsense mutation or bad through a missense. So both ways can give you
Okay, fair enough.
a faulty protein. Yeah, good. So what I'm looking for all of you saying here is basically the keywords I mentioned: ribosome, mRNA codon, tRNA anticodon. Sorry, Epidemiological, just do you mind muting yourself? Only because of the friction. Yeah, thanks. Good. So ribosome, mRNA codon, tRNA anticodon, and I want to also hear you mention that as a faulty protein was produced, this likely would have resulted in a missense or a nonsense mutation. Right? Good. And you can maybe be specific and mention 2 amino acids were affected here. So if you can put that into 2 sentences, you have all the buzzwords there. Okay. Good. Do you see how we're answering the question but also being specific to the question? I think that's what you're all missing, that little fine tuning where you need to answer the question and not just generic description of a process. Okay. Good. Micah, every time they give you data, what are you going to do to that data in the question?
Mention it.
Good. Not just mention, but try to put it in some kind of calculation. For example, if they give you 50 bases, you know it's 50 divided by 3 amino acids. So state the number of amino acids. That shows you understand what a codon is, right? Good. Describe the effect caused by mutation and the effect that this would have on the organism. Okay. So we kind of already spoke about that. The effect here is that it would have resulted in either a missense or a nonsense mutation. As a silent mutation would still produce the same amino acid sequence and still produce a functional protein. So what effect could this have in the organism, everyone? How would you explain this? I'm going to pick Maya. You have 2 lines to explain how these mutations would affect an organism. You can use 3 if you'd like, but give it a go.
A missense or nonsense mutation would result in a faulty protein being produced, which affects phenotype. Yeah, I'm not really sure.
Maya, you wasted 1 sentence because you repeated the question there. The effect it would have on the organism, right? And it says here it's a faulty protein. So you just repeated mutation, faulty protein, phenotype. So we're back at square one here. So Miriam, your turn. What would you say?
I would say the faulty protein could end up coding for a different process than what it was meant to do. Or if it was a nonsense mutation, it could end up with a dysfunctional protein due to the premature stop codon.
You know, both would result in a dysfunctional protein. One would just be more dysfunctional, but let's not focus on that. Now, again, I'm going to repeat the same question I've been asking you every single question because I think...
Can I have a go? I'm not sure this is right, but...
If you answer my question first, Bilal, which is what
Okay.
is this question asking you? Really? What is it really asking?
The effect it would have on the organism?
You guys are just repeating the question to me. I have eyes, I can read. You know, I have a functioning retina. Come on, guys. This question is testing your understanding of protein structure and function, right? So the question's told you you make a faulty protein. They don't want to keep on hearing, you know, the protein is faulty, the protein is dysfunctional, the protein is denatured. We've heard that enough times here. You need to explain how that's going to affect phenotype. Well, what do proteins do, Micah? What were the two categories we had?
Structural proteins.
Good, and?
Enzymes.
Yeah, functional. It doesn't just have to be enzymes, right? You're all thinking right now, do you know what neurotransmitter you're using to think, relay information, strengthen connections between brain cells? Right now, your brain, Maya, do you remember watching Harry Potter and the staircases that keep on moving?
Yeah.
The library, that's like your brain right now, especially at your age. Your neurons are wiring and firing and forming new connections and breaking old ones. Your brain is growing and changing, right? The reason I'm bringing this up is the neurotransmitter that allows you to do this is glutamate, which is a protein. Proteins have very broad roles. It's not just enzymes. It's not just enzymes for functional. They can act as neurotransmitters. There are protein hormones. You remember insulin, everyone? Insulin is a functional protein, right? It's an Uber, it's a microscopic Uber that takes glucose and drops it off into a cell. Good. So it's broader than that. So I want to just hear structural and functional. So you need to explain that the structural protein could be impaired, right? Or a functional protein could be impaired if the protein was a functional one. And you can provide examples there, right? And then you'd be answering the question, right? And you can talk about missense and nonsense as well. So you can mention a missense mutation may have occurred where one amino acid was altered in this protein, rendering it dysfunctional, or a nonsense mutation could have occurred whereby a premature stop codon codes for release factor and produces a prematurely terminated polypeptide chain. Then you need to explain the dysfunctional protein will affect phenotype as it can either serve a structural role or a functional role. Its structural role could be impaired, e.g., you can mention a structural protein actin myosin in muscle fibers, causing disease. If this protein were to serve a functional role, it would result in a biochemical change in the organism, e.g., inability to break down a compound, which can also result in disease. Give an example. Do we know any really famous examples of an enzyme and a disease it causes? Very famous one. It was brought up in your 2019 trials and it was on all the HC discussion groups and students were complaining that I didn't understand this.
Phenylketonuria.
Very good. Phenylketonuria, PKU. PKU or phenylketonuria. And Maya, do you remember what that reaction involves?
Not really.
So there is an amino acid tyrosine, and it's usually found in normal Coke. It's found in meats. It's found, I believe, in some milk. And it gets broken down into phenylalanine. And the way it gets broken down is OH groups gets taken off it. So we call the enzyme a hydroxylase. Those of you who do chem would know OH is a hydroxyl group. The enzyme here is called phenylalanine hydroxylase. Right? And you can have an autosomal recessive disease whereby you can't produce this enzyme because of a mutation, just like in this case here. And so what happens is you cannot eat tyrosine because the tyrosine you eat cannot get broken down anymore into phenylalanine. If it builds up in the blood and you eat tyrosine, you can cause damage to nerve cells. So you can cause mental retardation, seizures. And you typically see this in blonde Caucasian kids. And if they were a patient in front of you, if you're a doctor, one of the symptoms they might have, you might notice a musty odor. It's quite weird. That's what tyrosine produces when it builds up in the body. Yeah. Good. Okay. So you don't need to go into too much detail there, but can you always see why it's so important to know what the question is asking you? Right? You got to always, if you're confused, my tip to you is just pause and think, what dot point is this testing? So you can at least structure your answer to hit those dot points, get the marks. Make sense? Don't jump into it just blindly. Good. Why knowing your syllabus is so important. All right, next question. Recombinant DNA technology. Let's see. Okay, it's just loading on my screen. Can you all see it? How about I do? Okay. All right, here. DNA can be inserted into bacterial plasmids. What's a plasmid, Micah?
Circular DNA, usually in prokaryotic organisms.
So if I drew this in a diagram and I asked you 1 mark, identify this, and this was a bacterium, you saw the ribosomes, you saw the pili. What would you call this structure here, A? Identify this. What would you call it?
A plasmid.
Raiden, what would you call it?
Nucleoid.
Pardon? A nucleoid? Nucleoid. Interesting. Okay.
I'm just taking a guess. I don't know.
The thing is, I would say it's illegal to say anything about the nucleus of the prokaryote because they lack nucleus or membrane-bound organelles. Bilal, your turn. What would you call this?
Plasmid?
All of you would come out of your trial exam losing 1 extra mark. Why? Can anyone tell me why they were all incorrect? See, plasmid DNA is circle loops of DNA. You're completely right. But so is chromosomal DNA. A bacteria, all bacteria have 1 circular loop of DNA that is their single chromosome. Does that make sense to all of you? Right? So if you were to see a plasmid DNA, you should definitely be able to see the chromosomal DNA. Plasmid DNA is just much, much smaller. It looks like a really perfect circle band. Chromosomal DNA looks like a floppy hula hoop disc almost. You all see that? Right? So the answer there was this is the actual chromosomal DNA. Does that make sense, Micah? Plasmids are much smaller, they're circular, and if you can see them, you should be able to see the chromosomal DNA. This is important because 1 dot point in your HSC is distinguish the structure of DNA in eukaryotes and prokaryotes, and they love to test that. So it's very hard you'd remember this. So the answer is plasmid DNA are circular loops of DNA that are separate to chromosomal DNA. And what's unique about them, Bilal? Why do we care about plasmid DNA? It's a pretty cool effect.
Not too sure.
They can jump between bacteria, right? So if I was a bacteria, I could quite literally throw my little plasmid DNA across the room. Ishan could pick it up and he could take in that gene and express the trait, right? That's something we humans will possibly never be able to do, right? We can only transmit genes vertically. Bacteria do it horizontally through plasmids. Make sense? Good. Right, let's move on. To produce recombinant DNA, the diagrams show how a bacterial plasmid in its original form and the recombinant plasmid. Okay, I'm going to stop talking. I'm going to let you all absorb this because there's a lot of information here. Give you about 30 seconds to read it yourself as well. Sorry, I'll just delete the screenshot and so you can read the entire question. Okay. Okay. Now, this is a good question. So it sounds like a trial question. This is a very typical question that you'll get in your exams. Okay, so what would you do? First of all, you'd underline all the key terms, right? Because if you don't do that, then you're just losing 3 marks. So gene X, it's before and after. This is the before and after images of gene X being spliced in. Now, what's interesting that I note is that gene X has been spliced in and it's actually split apart gene B. Do you all see that? They wouldn't have done that for no reason, right? They could have just put gene X here. They didn't have to put it right between B. So my first question is, how's it going to change things?
It could affect the production of the yellow substance. So my guess would be the bacteria that doesn't have the recombinant plasmid would produce yellow substances as normal, but the bacteria that have been gene spliced wouldn't produce it.
Okay. But the original bacteria have this original plasmid. They don't even have gene X. So would they produce the yellow substance, Zachariah?
Oh, okay, they wouldn't. Yeah.
Right. So the first thing you think about
No,
is
wait. Gene X is for human hormones. Gene B is what codes the yellow substance.
oh right, yes. Good point. Good point. So let's highlight that.
So that's why it would break it, because it goes in between.
Yellow substance, gene X produces a human hormone. Good. So gene A and gene B could be mounted. So gene B is the yellow substance, gene A is your ampicillin, and gene X is your human hormone. Interesting. Good. Yes. So as you were saying that, so you were talking about the yellow substance, is that right? So you were mentioning that the recombinant, could you just recap
Yep.
that?
So what I said was, as you can see, the original plasmid does have gene B, so it would be producing the yellow substance. The recombinant plasmid, which is the bacteria that has been spliced, it has gene B, but it's been like interfered by this gene X, which would make it dysfunctional. So what you would like expect to see is the bacteria that hasn't got that recombinant plasmid would produce yellow substances, whereas bacteria that have been spliced, they wouldn't produce it. So that's how
Okay.
you'd like differentiate between them.
Very good. Very good conclusion. So you've perfectly answered the first issue here, which is that gene X has been put right in between gene B. So in effect, what's happened, everyone, is that we've caused a huge frameshift addition mutation to gene B. Can you identify that? A bunch of bases have been added right in the middle of gene B. So whatever gene B was meant to produce now, it cannot produce. Right? It's like putting a HSC textbook in the middle of an IKEA manual. The IKEA manual would no longer make any sense, and you would no longer be able to make a simple desk chair, right? So very good. You don't make whatever B is supposed to produce anymore. So you should, first of all, my plan would be that I'd want to identify that, right? So if you are recombinant, we've got a frame shift addition, and as a result, gene B would be dysfunctional. And what does gene B code for? Yellow substance. So there should be nil or 0 yellow substance. So this is how I would plan my answer. So I have a little flow chart of my thinking. So that's the first thing I would like to mention. Does that make sense to everyone? The next thing is I want to answer the damn question, right? The question is, how does A and B, how could we test for A and B? Now, what is A producing, Maya?
It's resistant to ampicillin. It codes for resistance.
Do you know what ampicillin is, Maya?
An antibiotic. I
Very
don't know.
You're completely right. Ampicillin is a broad spectrum penicillin. So it's a new and improved version of penicillin. You can think of it like the revamped Tesla. It's the ampicillin to the penicillin, right? We don't use penicillin anymore unless it's for specific bacteria like Streptococcus. So ampicillin is much better, and you might use that one day as well. Good. So what is this testing? I know I keep on asking the same question this lesson. Bilal, what are we testing here? For testing whether gene A is present, what syllabus dot point is this assessing here for you?
What syllabus dot point?
Yeah, you don't have to be specific to the dot point, but what general concept is it going to be testing for you?
Recombinant DNA.
No, not really. See, it's structured like a recombinant DNA question, but it's not really testing much about recombinant DNA. Do you guys have to mention restriction enzymes here? Do you have to mention electron
Oh
guns?
no.
No. So obviously it's not really testing your recombinant DNA. What is it actually testing? Come on, you've all seen these diagrams of petri dishes and antibiotics.
Or agar plates.
Yes, so straight away you should be thinking about that, right? We've got an antibiotic. We want to check if this bacteria, technically, any bacteria with this gene A, should be allowed to grow freely around ampicillin. There should be no zone of inhibition because they're completely resistant to ampicillin. Do we all agree? So if I get a petri dish and I put ampicillin right spot bang in the center, I would expect to see bacteria growing all the way to the margin of that antibiotic disc. There should be no zone of inhibition, as we call it, right? And that's what you see only when you have antibiotic sensitivity. We do this every day at hospital. We literally take a swab of patient's blood or patient's sputum or their pus sample. We send it to the lab. The lab will culture it in a petri dish and say, hey, these guys are resistant to this and that antibiotic, right? We do it all the time. So you need to understand how this all works. So here, there should be no antibiotic growing if the bacteria was sensitive, but we know the bacteria is resistant. So what would we expect to see if gene A was functional? If gene A is functional, bacteria, make a little thought loop in your head. If gene A is functional, then the bacteria are resistant. And if the bacteria are resistant, will there be a zone of inhibition? Good. Nil zone of inhibition. So Mike, is this not a decent test to confirm that gene A is working?
Yeah, it is.
Yeah, so that's what I'm asking. When I keep asking you the same question, it might sound stupid, right? What is this question asking? Well, obviously what's written on the page, but I'm trying to get you to think like a band 6 or a state rank student. Think one step ahead. What dot point is it testing? That's probably one of the hardest parts about trials. So if you can switch to that line of thinking, you'll do really well. Good. Now, gene X. We know gene X has to be working if it's been inserted as recombinant DNA. And that would render gene B dysfunctional. But how do we check if gene B is functional? Because that's what the question is asking. Explain how gene B could be monitored. So up here, everyone, do you realize what we've actually done is we've identified why we should monitor gene B. We should monitor gene B because according to this extract, whatever they're doing in this extract should render gene B dysfunctional. So you want to see if it actually worked out. If gene B is dysfunctional, everyone, what does that mean about gene X? Can anyone tell me the conclusion there? Very important. If you note that gene B is dysfunctional, what can you say about gene X?
It's been spliced in the right place, so it will probably be functional.
Yes. Do you all realize the only thing that would cause gene B to be dysfunctional is the addition of gene X? So we actually know that gene X has been correctly inserted into the plasmid when gene B starts to become dysfunctional. Do you all realize that? Very important. That's why this whole question is written there for you. Good. So
can you go over that again one more time?
Yeah, sure. Sure. I think below the way you should be thinking about this is don't think of it as plasmids. Plasmids are confusing. What I would do in my HSC is I'd draw two bacteria, right? I draw one bacteria here and I draw another bacteria here. Right? So what they're doing is they're inserting gene X. Do you agree, Bilal?
Yeah.
Let's call bacteria 1 bacteria A and this bacteria B. Okay, and I'll take the A out there. Okay. Now, Bilal, how do we know whether we've put gene X in correctly? Because gene X just makes a yellow substance. You might be able to observe it on a petri dish, but how can we confirm whether gene X has been put in? When gene X gets put in, what gene does it disrupt, Bilal?
Gene B.
Yeah, so bacteria B can no longer produce the protein for gene B. What does gene B code for?
The yellow substance.
The yellow substance.
Right.
So now it should no longer be able to code for the yellow substance, right?
Okay.
So we can say that it's gene X that is causing gene B to not be expressed and then code for the yellow substance. That's what we're saying here. That we know that gene X has been correctly inserted when gene B stops working, full stop. Yeah, good. Good. All right. So coming back to this, gene B, how could gene B be monitored? I kind of gave it away.
The
Same
presence
thing,
of the
guys.
yellow substance, or if it doesn't produce.
Yeah. So how do we confirm? So we can't just look at a bacteria and say, it's making yellow substance, right? How do we confirm whether we're making mass amounts of yellow substance? Same thing as A, except we're not going to put an antibiotic disc. What are we going to do? Miriam, tell me.
If you put like a control group or something?
Oh,
and
Mary,
compare.
you're confusing it. You're complicating it. Simplify it. How do you know? Anyone? Alec, how do you know? How do you know whether gene B is working or not?
Can you compare it to the original plasmid?
Yeah. You all got it. I'm pretty sure you all not. I think you're just not saying it because it's so obvious. Let's start over. If I say a bacteria makes a yellow powder, how do you confirm that the bacteria does make a yellow powder? What would you do?
If you see a yellow powder,
Where? That's what I'm asking specifically. On a
petri dish.
Agar plate.
yes, that's it. All I wanted you all to say was swab the bacteria on a petri dish, incubate it at 37 degrees for 2 or 3 days, and after that, look at the macrocolonies. If they look yellow, you know that gene B is working, which means gene X was not put in. And if gene B is not working, you know, well, congratulations, you are now making whatever hormone that gene X codes for because it's correctly been inserted. Does that make sense to all of us? So to get the remainder of the marks, your last mark would be, see how it's just so systematic. Your last mark would be 1 mark, student identifies that they would have to culture. This is for gene B. This is for gene B. You want to culture it, you want to incubate it, and you want to observe for yellow color. And if there's no yellow color, no yellow, that means gene B is not working, which means, therefore, gene X has been inserted. Does that make sense? Very good. Any questions, please do ask. So if I were to ask you all again, what was this question testing? It was actually everything but recombinant DNA. Do you all notice that? It was testing your understanding of antibiotic discs and zones of inhibition, so the efficacy of antibiotics. It was testing your understanding of the antimicrobial testing of water samples. Do you remember that experiment you all did with Petri dishes? Right? And it was testing whether you remembered how to use that. So it's interesting. You all bring up very good questions. Do I need to talk about BT cotton? And do I need to talk about E. coli, transgenic E. coli? Not at all. So do you see how this question has nothing to do with recombinant DNA? Yeah. Good. No questions at all? Okay. Very good. Good job. Who put this question up? Good job.
That was me.
Good job, Bilal. All right. So, Miriam, your question now. Biotech. Oh my, that is a big question. How about this? We've got a bunch of long responses. Actually, no, it's the same one. 9 marks, 9 marks. We'll come back to the 9 marker. Just remind me to someone jot it down that we need to come back to the 9 marker. Miriam, it's your question, so jot it down. It's gonna take us a little bit. I want to go through the shorter ones very quickly. What do we have here? Adaptations of 2 pathogens that facilitate entry and transmission between hosts. Easy, we can do this one. It's either you know this or you don't know this. You can't really make too much up. So, come on. What are your 2 examples? Let me ask Serene, give me 1 example. Pick a pathogen and tell me 1 adaptation it has for entry or transmission.
For virus, they contain the
Virus, yes?
yeah.
What do they have?
They contain a capsid protein to protect them from being destroyed by.
Protects against. I'd say protects the genetic material. Okay, protects the genetic material of the virus. Okay, protects genetic material from host immune system. Facilitating transmission. Facilitating transmission. Now, what I would say here, everyone, is this is okay, but it's very weak. Do you know why this is a weak adaptation? It's because all viruses have this. So yeah, you could talk about this. You could, but it's just very vague. You want to be specific. What are some specific adaptations bugs have?
Vectors.
Pardon? Raiden, a bit louder?
Oh, I said vectors, like mosquitoes can bypass the skin barrier by using pathogens to inject into the blood directly.
Do mosquitoes need to use pathogens, or do pathogens need to use mosquitoes?
Mosquitoes inject the pathogen into the blood.
Yeah. So whose adaptation is that? Is it the mosquitoes' adaptation?
Mosquitoes adaptation.
Well, we're talking about pathogens here, so not vectors.
Could we talk about like enveloped viruses and non-enveloped?
Very good. Any idea how? I'll give you a little hint, right? Hep A and B are enveloped viruses, which means they have a phospholipid bilayer, which means they're posh, comfy, and sensitive viruses, right? They can't travel through the digestive system. If they do travel through the digestive system, the phospholipid bilayer gets destroyed, the virus gets destroyed. And so those viruses can only be transmitted sexually or through blood. But Hep A and Hep E, they are non-enveloped, everyone. They don't have that posh, soft glycolipid, phospholipid bilayer, right? They, since all they have is a protein capsid, they have no envelope or outside of that. Those viruses can pass through the harsh pH of the gastric acid, the alkaline conditions of the small intestines, and they tend to cause diarrhea, vomiting, and a gastroenteritis kind of illness. Do you all see how that's an adaptation? So not having an envelope is an adaptation. Did you all understand that? When you don't have an envelope, right, you don't need to protect or save that envelope. And as a result, you can pass through more harsher conditions. And do you see how this is very specific to transmission? This one here is just saying, it's got this capsid which enables it to transmit. But what we're saying specifically here, it's still about the capsid, but we're mentioning specific diseases too. So Hep A, for example, e.g., is a non-enveloped virus. So this is the same, I'm just improving this one here, right? It's a non-enveloped virus, which enables it to protect viral DNA against gastric acid, alkaline intestinal secretions, and it can cause, therefore, fecal-oral transmission. Fecal-oral transmission. Do you see how now we're specifically linking it to transmission? So I'd say this is a better answer. You'll see how it's just an improved version of what we said above. So I'm going to rub out this section here. Make sense to everyone? Good. So capsid, tough protein capsid protects the DNA. Tough protein capsid. Very good. What else do we have?
Plasmodium falciparum.
Yeah,
So
what about
that's
Plasmodium?
So it's a plasmodium, and basically it's got an extreme or like a very difficult life cycle, which allows it to get past like most of the immune cells, which would otherwise defeat it. So that allows its entry into the host.
See, that's a very good one, but you need to be specific about the life cycle. Do you remember anything about the life cycle, Zachariah?
Yeah. So basically, the plasmodium will enter into blood cells themselves, and therefore they go in like undetected in the blood vessels, and yeah, from then on they will just replicate, burst, and then go into other blood cells.
So yeah, so thing is in med school, I remember 2 years ago, and even now I have to relearn everything that I've learned in the past 5 years. We have to memorize the life cycle of malaria and the Plasmodium pathogen. And the interesting thing is this pathogen sits in your liver, usually, and it's in a dormant state. And I think of it's like in a state of hypnosis. It's dormant. So we call those hypnozoites. That's the name, hypnozoites, right? And it remains dormant there, and we can't pick it up in the blood. It causes no symptoms. And so someone can have malaria, come back to Sydney, Australia, be completely fine for up to a month. And then suddenly they can break through fevers. And the interesting thing about malaria is the fevers are the plasmodium then goes to red blood cells.
Prithvi, can you repeat? You kind of got cut off before. Can you repeat
Which
the last
part?
part?
Yeah, okay, I will. Good. So all I was saying was that malaria can be stored in the liver in a dormant stage, and it can infect red blood cells from the liver, travel in the red blood cells, and it can burst the red blood cells, right? And the bursting of red blood cells triggers the immune system, and that can cause your systemic responses like fever. So it bursts, since it has a very cyclical lifestyle, right, where it forms in the liver, it's dormant, and then it infects red blood cells, bursts red blood cells, and then infects more. You see a cyclical pattern of fever in patients, right? So malaria can cause a fever every 4 days or so. And so here it goes in red blood cells, and we have another stage called the sporozoite stage. And the only reason I'm telling you this in the context of your exams is Zachariah's example was perfect, but he needs to mention buzzwords, at least one, right? So he knows what he's talking about. So when you say they have a complex life cycle, you can mention that Plasmodium falciparum is a protozoan that has a complex life cycle consisting of dormant pathogen in the liver, and we can mention sporozoites that are in the red blood cells. And this enables it complex life cycle. Yeah. So it's multiple stages. So technically the stages here, this would actually be called gametocyte. Then it will become a sporocyte, but you don't need to know that level of detail at all. Okay, as long as you mention one of these buzzwords. It has complex life cycle in which it travels between the liver and red blood cells. I think that will be enough to give you full marks. So maybe just mention liver and red blood cells. Don't worry about the buzzwords there. And this enables it to evade host defenses. Good. So those are all of yours. Now, the ones I usually think about are this one here. This is a really good one. You can mention prions, right? Prions can invade dendritic cells and thus travel via autonomic nerves from the digestive system to the brain. So that's how prion diseases can go from the food you eat. So the people who are cannibalistic and ate their deceased relatives with kuru, that's how this prions traveled from the digestive system to the brain. Now you might ask, well, what the heck are autonomic nerves? Has anyone had adrenaline course through their body, maybe through when they did an arcade roller coaster, or you might have had a life-threatening moment where you saw an animal or got confronted, right? If you remember, your pupils dilated, the hairs on your body rose, blood rushed to your muscles for the fight or flight response, that is all triggered by your autonomic nerves. So the way I think of it is autonomic nerves serve automatic functions in the body. So your rest and digest and your fight and flight. Okay, so that's all that is. So that's the nerves, the specific nerves that prions will invade to travel to the brain. And do you see how this is facilitating their transmission? Right, so that's one I have. Another one you can remember, it's a simple one, is bacteria. And please do not remember all of these. You're just wasting memory space. Pick the ones that works best for you. I'm just giving you a ton. Bacteria can have enzymes like hyaluronidase, A's because it's an enzyme. Does anyone know what hyaluronidase is? is particularly for the people that use skincare product. Does anyone know why people like to use hyaluronic acid on the skin? Anyone seen K-pop actors? They love to use it because it gets the skin nice and fair and plump. It glows your skin. So it's a part of your connective tissue. So when you add it onto your skin, it's meant to plump it up and make it, and it technically does work, right? So hyaluronidase, it breaks down the hyaluronic acid. So it allows the bacteria to invade through your tissues. Acid, and therefore you get increased invasion. I'm not going to give you any more. This isn't a medicine class, but you understand the basics here. Pick whatever's simple. I would probably not pick plasmodium just because it's a little bit complex. I would probably go with this, and this one's easy. Just remember one enzyme. Good. Any questions? Okay, that was not too much learning. It was just concept, and concepts are not what you should be focusing on now. Let's move on. Okay, I know I'm just jumping through questions, but I wanted to go through the application ones first. 30 seconds. Raise your hand once you've got an answer, and then I will give you a break. This is so relevant to your lives right now. Schools, New South Wales Health put out a message, not health, but Gladys and the government mentioned that there's no confirmed return to school date now. Is that right? So the 16th might not even be a return date. Is that what you've all?
Really? Wait, can you hear me now?
We didn't know that.
We were confirmed that
Yeah.
it's 16th, but it might not be.
I don't know. Check the news. But I just saw
Can you guys hear me now, Bart?
Yeah, I can hear you now, Alec.
Yeah.
Oh, yes. I switched over to my phone because I don't know what was happening with my laptop.
That's okay.
Sorry about that.
No, that's fine. Yeah. So as I was saying, yeah, just stay tuned with the news. I don't know the specific details, but yeah, it's a weird time. And the public, some people are very upset that all of you are getting the Pfizer vaccine before them. That's interesting too.
Our teachers are mad about that because they don't get priority apparently. And then
Your teachers don't get priority.
Yeah.
Oh, okay.
Yeah, they're not counted as essential, I think, or as even though they probably should be.
They should be, but makes
No,
sense.
but see, technically, when you're sitting a trial exam, your teachers won't be at school. It'd only be these. It's usually with the trials, it's dependent. With the trials, they should be. With the actual HSC, it'll be at your school exam halls, but there'll be completely random old people who've just signed up to be moderators for the HSC exam. So I don't know, but anyway, we'll come back to this. This is all so logical. So you have a breakout at a school. Just think about this is happening in Sydney tomorrow, right? There's a huge breakout at a school, right? And we're trying to, and we do not want it to spread to them, right? Maybe we'll do red for the infected people. Would we want people from that outbreak area to move out, whether it be teachers or students, right? We'll look at B. Staff. Would you want them to move out to obtain antiviral medication?
No.
No. I don't like the out part. To send staff out. No. Antiviral medications can be shipped to the facility, but they should not move out. So the issue here is sending staff out. I don't like that at all. What do we think about A? Maya?
I chose A. Yeah.
Okay. I would first of all say, it's not practical. If this actually happened,
Yeah, I was going to say that.
Yeah,
no one will keep you at school. So you just keep your logic hat on. I just mentioned it's impractical. It's honestly not a good answer.
It
Right?
could be faulty too, because if they stay at it's to stay at home, right? To keep to keep them at school.
But think about this. Only some people have been infected. Imagine keeping everyone at school, and now they have to be exposed, the uninfected people as well, with the infected people. So do you see the issues with A? Right? Yeah. But we have to keep moving on. So I say B is definitely not right because we are not letting people move out to unaffected areas, right? Okay. C, to disinfect all eateries. Okay, well, we know measles. If you may not know this, but measles is caused by a virus, right? So it is a viral disease. But disinfecting all eateries, desks, and recreational areas. But is that going to stop person-to-person transmission?
No.
Do you see with COVID-19, are we moving out on train stations and wiping the trains vigorously? Is that our prevention strategy? No, not really, right? So it doesn't make sense when it's a person-to-person disease to just sterilize the environment and say, thumbs up, we're good. So I think C is definitely a horrendous answer. So B and C are horrendous. A is just shoddy. What about D? To bring in medical officers to vaccinate the people in school. Now, superficially, it looks like a really good answer. But actually, it is not. And can anyone tell me why?
Because the question asks how to prevent it from spreading to the rest of the state.
Yeah. And if you're infected, see, if this is you and you are infected, we don't jab you when you're infected. We don't, if someone has COVID, we don't go, here, have the Pfizer before everyone else. There's no use because their body is generating active acquired immunity in that process. There's no need to give them the mRNA for a COVID spike protein. Does that make sense to all of us? In fact, you could probably argue, this is a good exam question. It could say, person A has suffered from measles and they've survived. Person B has been vaccinated with measles. Compare their immune systems. That's a very good question, because then you can be like, okay, well, person B, if they were vaccinated, could be passive immunization, active immunization. And then you could compare the two to the diseased person. But what I'm trying to get across here is if you have disease, you're not getting vaccinated, there is no point. It is useless. So this is a useless measure to use. In fact, we would want to vaccinate everyone here so that even if someone spreads out, they will not have anyone to transmit it to. Does that make sense to all of us? That's what we're trying to do in this pandemic. So A looks horrendous, but B and C and D look completely unselectable. So by logic, what's your best answer here, everyone?
A.
This is why trials are hard. Trials are not hard because they'll ask you some fancy, stuff that you just haven't studied. You would have definitely learned it. Is they're going to ask you crap questions, and you just have to put your critical thinking hat, pick the best answer. Okay, so if you're stuck, yeah.
I was confused because when I did this question, I put A as well, but then the answer said D, so that's why I was confused.
D is completely wrong. It makes no sense. You can definitely talk about this in the trials.
Yeah,
But
okay.
you can literally bring out our current program to prevent COVID-19, right? When we don't vaccinate when there is an outbreak in a school, because in a school, half the people are infected. What we would do is we'd quarantine everyone who's potential exposures. We'd take everyone who's safe. We would isolate and quarantine everyone who's infected and treat all the infected people. And we'd vaccinate everyone in the surrounding area. That would be the best plan of action. Does that make logical sense? Yeah? If you have infection, you don't vaccinate. And if your potential exposures, you need to isolate first to confirm you don't have infection, right? Let's say you're in this school, you get the disease. Let's say I get the disease, I'm at your school. Mike is a potential exposure. We would not straight away jab Micah. We'd actually isolate her, get her to self-isolate for the incubation period, confirm she's negative, then she will be eligible for the vaccination. Does that make sense to all of us? The vaccinating is not the plan. This is, yeah. Sometimes the people who write these papers, you have to realize they're also teachers. Sometimes they don't know the best practices for science. A bit worrying. Okay. This is a very good question. Thank you for putting this one up, Abhinay. We'll come to the 9 marker. All right, before we do this, I'm going to let you all take a bit of a breather. So, would you like a 5-minute break? Can you all raise your hand if you would like a 5-minute break? Okay, sure. Make sure if you are taking the break, stand up and stretch. If you're sitting, it's no use. Otherwise, your brain will fall asleep. All right, I'll see you all at 8 o'clock. So stand up, stretch, get some water. Feel free to put up any additional questions and we will continue. Good. And thanks again for all of you for putting these questions in the chat. Very good. You learn the most this way. Okay, I'm gonna get some water too. All right. Let's jump back into things. So when you've all returned, feel free to turn your camera on. We'll jump right back into it. Anyone doing anything else on top of their HSC workload? Anything interesting?
Binging a bunch of anime and reading manga.
That's good. That's good. I remember I think I was watching Attack on Titan when I was in year 12. I don't remember. But no, maybe I was reading it. I don't remember at all. But yeah. Brayden has a book going. Why We Sleep.
Oh, it's good. There's a lot of good stuff in there. A lot of like been educated on a lot of stuff about sleep, especially like REM sleep, non-REM sleep, importance of it.
Yeah, it's very interesting.
Pretty good stuff.
Right?
Yeah.
You get all of your basically the benefit of sleep in those 15-minute REM cycles. It takes like 90 minutes to even get into one, and you only have 5 or 6 REM cycles. And I guess the interesting application there is what happens is if you're all sleeping and then you hit the snooze button and you sleep for 5 more minutes, it's actually worse than not sleeping in, right? Much worse, because what ends up happening is you start a REM cycle but you don't finish it, and so you wake up even groggier than you would otherwise. So it's quite interesting phenomenon.
Had a lot of experience, a lot of experience with that as well. So
I'm sure all of you have. I do pretty much every day. Anyway, we'll come right into this one. So, how important is it for genetic material to be replicated exactly? Consider the life cycles of both a pathogen and a host, using at least 2 examples from infectious diseases that you have studied. I think if I was a teacher and I was to write a trial question, this would probably be the question I would write, because I've thought about this so many times and I was waiting for them to give you a question about this. So I'm not going to say anything. You'll have 2 minutes to create a plan. 2 minutes. Now, I'm probably going to stop. No, I will keep screen sharing, but I'm going to make my own plan, maybe in my head. Okay, I'm going to start butting in and picking on people. So you've already started your plan. So Maya, what's the first point in your plan?
The judgment. So I said it wasn't important.
Interesting. So it's not important for the pathogen and the host?
For the pathogen.
Okay. Okay. Now, remember, Maya, do you only have to talk about the pathogen, or do you need to talk about the pathogen and the host?
The pathogen and the host.
Does anyone know why you need to talk about the host? Let's say you're on the fence right now. You're in this exam, you're thinking, do I need to talk about the host and the pathogen or just the pathogen? What would tell you need to talk about both?
And
I mean,
It
doesn't
literally
it
says
say it
both.
says
Yeah, good. But any part of the syllabus? There is a literal inquiry question in Mod 5 which asks you, how important is it for genetic material to be replicated exactly, word for word? So if you remember that, you would know, I do need to talk about host DNA replication, DNA synthesis, the enzymes involved in those processes, because that's what that's the dot point that would also be tested here. Does that make sense? Good. So Maya gave a judgment, but I would fine-tune that judgment to say it is very important for the host, which is usually humans, and it is it important, Micah, for the pathogen?
No.
No, why is it not, Micah? Can you just give us a dumbed down answer why?
Because it's able to, it doesn't, if it gets mutated, it might actually make the pathogen more powerful, like, so it's not like that important for it to be replicated exactly.
Good. Now, Raiden, why would you be worried about mutations? If I got a invisible gun and I gave you 20,000 mutations right now, what would you be worried about?
how it affects me.
Yeah, more specifically, what would you be worried about? Give you a hint. I'm currently in my oncology rotation, and you should be worried about the disease that oncology studies. The C word.
Cancer.
Come on. Cancer,
Okay.
yes. You don't want to get mutations because that can lead to cancer and death. But the question here is, Serene, are viruses living?
On non-living things.
Yeah, then they're on the brink of living life and death. It's interesting, but yeah, you could regard them as non-living. So if you're not living, you're not worried about diseases like cancer. You're not worried about death. So if anything, mutation is good because it allows you to build populations that are resistant to selective pressures, which in this case are drugs to the pathogen, right? So my point here would be that's very important for the host, but it's not important for the pathogen. The next thing I would define is what is genetic material. You need to underline these words. So Zachariah, what genetic material are we talking about?
For the host, we're talking about DNA, and for the virus, it depends. So it could be DNA or it could be RNA, mRNA.
Good. Very good. RNA. Good. And what would you like to explain now? So you've identified the genetic materials, and what else would you like to identify? So with a host, how would you describe the life cycles of DNA replication in a host? That could give you a mark.
You could talk about how part of the cell life is to replicate its DNA for cell replication, and briefly explain that mitosis is DNA replication is a process for mitosis, which basically allows an organism to heal, the host to heal. It also allows growth, and it allows it to grow to gestational maturity. I guess that last point doesn't really matter. Yeah.
So I'd say the buzzword I've heard from you so far is mitosis, but I'm looking for more buzzwords. So now we're talking about DNA life cycle in the host. So who wants to explain that to me to see whether they're going to get full marks for that point? Bilal, your turn.
So I was thinking the two daughter cells must contain like the genetic information in the two daughter cells will be corrupted if the mutation was to happen.
Come on, guys. I'm asking you for the life cycle of DNA in the host cell. You just explained to me why it's bad in very general terms, that it's not replicated exactly. What I'm asking here is how is it replicated exactly? Explain that to me. Micah, your turn.
Like just the DNA replication process?
Yeah.
Oh, so like helicase unzips the DNA.
First of all, when does this happen in mitosis? That's what I want.
Oh, in interphase?
Which part of interphase?
The S phase.
Very good. S for synthesis. So S phase of interphase, we have such
I
as
think I
helicase.
mentioned that when I was explaining.
Oh, good. Did you mention the interphase?
I didn't say interphase, I said S phase. So yeah, I missed out on that.
Okay, good. Yeah, so S phase of interphase, we get the DNA replicating, and it's done through helicase unzipping and unwinding, primers and polymerase attaching complementary nucleotides, and ligase strengthening the sugar phosphate backbone. Full stop. You see, in one sentence, you can explain the entire methodology of DNA replication. Right? Good. So you'd want to identify that. So I'd probably say 1 mark for mentioning the process of the DNA life cycle, so the genetic life cycle for the host, right? So mention it occurs in mitosis, and then mention, where in mitosis, and then mention the enzymes and processes. Right?
Yeah. Wouldn't you have to mention polymerase as well? Or?
Yeah, so what I said then was I said we use helicase to unzip and unwind the backbone, and primers and polymerase construct the complementary strand. So that's how you can add it. Okay. And then ligase will strengthen the sugar phosphate backbone. Very good. Okay, so that would probably give you at least a mark. Now, where will we get the remainder of our marks from? So I'd say 1 more mark for Bilal's point, but I think you need to be more specific, Bilal. I need to mention if DNA is not replicated exactly, we result in mutation. Mutation can lead to disease by altering the protein produced, and it can affect processes and cause cancer, etc. Right? Which can lead to death etc. Right? So what I would mention here is inaccurate replication equals mutation equals to dysfunctional protein, altered phenotype, and disease, cancer, and death. Okay, so now I've explained the life cycle for the host. I've highlighted why it's important. So I've provided points for why it's important. So I think I like what Maya did. She assumed this was an assess question. I think she was looking for a verb, couldn't find one because this is a trial question and sometimes they're worded funnily. But you can structure it like a judgment. So this is your judgment. This is your points for why it is important for it to be replicated exactly. And now we're going to provide points against. So points against, which is why the pathogen's genetic material does not need to be replicated exactly. Let's structure it the exact same way. How many marks have we hit? About 4 marks so far. Right? About 3 to 4 marks. So we have about 4 marks left. So I would structure it as 2 marks for one pathogen and 2 marks for another because we're using 2 examples. Do you all agree? So what 2 examples shall we all use?
Can we use like the influenza virus?
You can, definitely. I think influenza is an amazing example. It's such an amazing example because a virus mutates every single year so much that we actually need to create a new vaccine for it, looking at the winter of the other hemisphere of the planet to predict the strains that will affect us. So when it's our summer, it's winter in the US, right? Christmas Day, US winter. So they have their peak influenza cases. And so genomic scientists, they share the data about the influenza strains, the antigens, etc. We gather that data, we construct mathematical models using coding and data science, and we predict how it would mutate once it gets here in June, July. Quite interesting. And that's used to make a vaccine that saves thousands and millions of lives, right? Influenza kills. It kills the very young, kills the very old. Good. Do we know how many percentage of people die from influenza? COVID is about 2 to 3%. What about influenza? All of you will get influenza about once a year on average. It's important to know this. What was your chance of dying the last time you had it? You could have died. 0.1%. About 1/10 that of that 1/10 of COVID. Okay. Good. All right. Coming back to this, 2 marks. We're going to pick influenza. I think it's a very good example. So influenza, we need to first explain the life cycle before we explain why it's not important for genetic material to be replicated. So what is the life cycle of influenza? Micah, you picked it, so you must explain it. What kind of virus is influenza? Is it an RNA or a DNA virus?
RNA?
Yeah, so influenza is an RNA virus. You didn't need to mention that, but it's good to know this stuff, right? Especially with your exam answers to reach a band 6 level. So what would you like to mention, Micah? You have 2 lines max to explain the life cycle of influenza. Go.
So it infects the host, but over time it can develop mutations through errors in genetic material being replicated, which
is that part of its life cycle, Micah? If you're talking about the immediate life cycle of virus, are you going to start talking about mutations straight away?
I guess not.
Probably not, right? Because the virus itself, think about COVID, right? When COVID infects a person, it's not mutating actively there. It mutates over longer periods of time, hence we reach new strains like the Delta strain. But every time it infects someone, it doesn't mutate. And the life cycle refers to when it infects a cell to when it pops out. Does that make sense? So you usually don't get mutation right then and there. So let me ask, who haven't I asked so far? Ishan, been quite for a while, so give it a go. Life cycle of the virus.
So for influenza, it starts by entering the host. And then it has to express its genes and replicate it in order for it to increase in numbers, and then so all I have at the moment.
Okay, I'm going to make it very easy for all of you. You're going to get 1 mark if you explain the life cycle. You're going to get 1 mark for if you explain why genetic material is not important to be replicated exactly. And then for pathogen 2, 1 mark for the life cycle, 1 mark for your explanation. So you see how we've broken it down so systematically? So if you hit all these points, you're going to get 4 marks. Now, Ishan, you wouldn't have got the 4 mark there for life cycle of a virus. Come on, this is so important. You're in the COVID pandemic. You need to know how the virus works. Question, Micah?
Can I try again for life cycle?
I'll let you think about this. I'm gonna give it to one other person. I'll get Abinane. I haven't heard you talk for a while, so give it a go. What would you say? Micah will save you if you don't get this right.
Doesn't it just like hijack the transcription translation of like a cell, and then it bursts out afterwards? Like it produces viral proteins within like a cell, I guess, and then
Okay.
after that, something.
So yeah, you'd get. I would want to give you half a mark there because you definitely hit 50% of the answer, but you're missing the other 50% of the answer. I think it's kind of what Ishan said, but we'll talk about that. So Micah, your turn.
So they enter into a host organism and they use the host enzymes to make copies of the cell, like with the virus's virus's RNA in it, and then like this is able to infect the host.
Oh, we're all getting 50-50s. Come on. Maya, your turn.
Wait, so could we say like the antigenic drift shift stuff for how it enters the cell?
Where did you learn this, Abinane? Antigenic drift and shift?
Like that's my example for viral entry into a host. So
Viruses don't enter by antigenic drift or shift. So for everyone who's wondering what the heck shift and drift is, so you'll remember in 2011 when swine flu came out of nowhere, right? So shift occurs when 2 different influenzas that are completely different meet and they have a baby. And the baby virus is completely new. It's got a mix of one completely different subtype of influenza and another completely different subtype. It's very rare. It only happens in one organism that's infected by 2 different viruses, right? So shift is very rare. When shift happens, you get a pandemic. So swine flu was the last time shift happened. A pig influenza and a human influenza from a pig worker combined, and it caused the nasty hybrid that killed off a ton of people. So shift, it's rare. Drift happens all the time. Drift is what's caused COVID new strains, everyone. So the new Delta strain of COVID was caused by drift. Drift happens just because a virus is non-living. It doesn't have any checks into its DNA. When a virus copies its DNA, unlike us, it doesn't care whether it's made the exact copy or not. It's kind of like a drunken person. If you get them to copy a page, half the words are going to be different, right? That's what a virus does. And so it doesn't care. And so every time the virus replicates, its genes slightly change. And so we call it drift rather than shift because it's slowly changing. Forget about all that for now, but you are technically talking about drift. That's not till the life cycle again, Abinane. That's when you explain the effect. So that's why I've taught all of you that just now. What I wanted you all to say was this. A virus's life cycle consists of 2 things. You all are missing one or the other, so get a red pen and write this down. The first thing is it wants to replicate its own DNA. The second thing is it wants to produce viral proteins. So Abinane, when you explained it, you didn't talk about viral DNA replication, and I believe Micah, you might have mentioned the DNA replication, but not necessarily the protein side of things. So what I would want to hear, this would be a perfect answer. Virus uses surface glycoproteins for entry into a cell. It will then use viral enzymes to reverse transcribe its RNA into DNA. The DNA will then be used via the DNA replication pathway using human helicase, polymerase, and ligase to produce more viral genetic material. Contrastingly, the viral RNA will travel to the ribosome for direct translation into viral polypeptides. New viral particles will then be assembled from the newly constructed genetic information and polypeptides. Does that make sense to all of us? It doesn't need to be that clean, but as long as you mention that two things are happening, there's DNA replication and there's translation of the viral RNA. So guys, the reason that viruses have RNA instead of DNA is because it makes the job easier. Do you all realize that? The RNA just has to go straight to the ribosome, make protein. There doesn't need to be any need for going to the human nucleus and causing transcription. It's kind of like a fast track for the virus. Make sense to us all? Everything should logically make sense in biology. Don't just memorize stuff. Good. So once you've explained the life cycle, what I wanted to hear for all of you was the DNA replication and the protein synthesis. Make sure you spoke about that. The effect is, as viruses are non-living, they do not have error check mechanisms for the DNA. And so we get antigenic drift. If you'd like to mention that buzzword, you can. You will not get a mark for mentioning that, but it'll look really good in your answer. So they are prone to antigenic drift. Why is that beneficial? You want to explain that. Michael, why is it beneficial to have antigenic drift? Two reasons, actually.
I don't know.
You evade the host immune response, and you evade pharmaceuticals, such as antivirals. You can grow resistant to antivirals. So those are the two benefits of a virus not replicating its material exactly. Does that make sense to us all? Very good question. Now, I'm done spoon feeding you all. Give me a second pathogen. Tell me its life cycle and tell me how it's important or not important for genetic material to be replicated. I'll pick one for you all, bacteria, because the others are a little bit too complex. If you have to talk about life cycle of a fungi, oh, fungal diseases. Even junior doctors wouldn't have a good understanding of that. So don't jump into all of that. Let's talk about bacteria, which are much more common. So what are we saying? What example do we have of a bacteria? Pick any bacterial disease.
Whooping cough.
Pardon?
Whooping cough, isn't that bacteria?
Yeah, you are right. It is a bacteria. Whooping cough is caused by, do you know the name of the bug?
No.
Bordetella pertussis. Bordetella pertussis causes the disease pertussis or whooping cough. It releases toxins that irritate your trachea so much that you'll cough. And the characteristic pattern is you cough so much that you keep on exhaling, and at the end of all your coughs, you inhale, and hence the whoop of the disease, whooping cough. Does that make sense to all of us? Yeah, it lasts for ages. Take antibiotics, right? So what's the life cycle of a bacteria, everyone?
Binary fission.
Yeah, very good. It replicates via very good. Very good. The reason I'm saying this is because he's linked it to mod 5 now. And it's very important that you see for an 8-mark question that you link multiple modules. Good. So it replicates via binary fission. Keep going.
Like what happens in binary fission?
No, life cycle. You could explain a bit more about binary fission. So I'd say the cell elongates, DNA replicates, and a cross wall forms. Cross wall is when the new peptidoglycan cell wall forms around the remaining 2 daughter cells. Okay, what else can we mention? We get invasion and secretion of toxins. That's part of the life cycle too. Invasion of bacteria and release of toxins. Those are the two ways bacteria cause disease. They either will release a toxin. In the case of Bordetella pertussis, you release a toxin, and they can also invade through tissue, right? If anyone's had a pimple before, what actually happens, the reason why you get a pus at the top of the follicle is because the bacteria and the neutrophils are releasing enzymes that are breaking through your skin and traveling to the skin surface. So it's literally liquefying your skin, traveling up to the surface. So it's invasion there, right? So the effect, why do we not care for bacteria about preserving their genetic material? I'm going to ask, and I want an exam answer here. So Alec, give it a go. Since your mic is working.
Well, we don't want to preserve. Wait, why we don't want to, or why we would want to?
Why we don't want to preserve a bacteria's genetic information.
Well, we wouldn't want to preserve it because if that bacteria stays within the body, that it may end up within the DNA strand.
Well, remember, Alec, you're thinking about the bacteria itself. We're not talking about the host anymore. If you're a host, you don't want bacteria in your body. But think you are a bacteria. You've been reincarnated, you're a little bacteria right now. Why would you not care if your genetic material changes?
I'm not quite sure.
Because there's so many of you, right? Bacteria don't care about me. They care about causing disease or not causing disease. So there's so many and they replicate so fast that if one bacteria dies, that's fine. Another bacteria will take its place. But the net effect is, if you keep on changing your genetic information, you will increase variation in the population. Do you all agree? So no matter what drug you throw at this bacteria, there'll always be a small subset who will be resistant, just due to mutation and variation. And that's how bacteria keep on surviving in this world. Make sense? They survived the meteor crash of the dinosaurs, right? The dinosaurs all disappeared a few 100 million years ago. Bacteria survived, right? And it's because they're so varied and they can survive such varied conditions, right? Very good. So key thing you need to mention here in your exam answer, maybe I'll write it for you, a little sample one, is mutation is beneficial as it increases genetic variation, and you link it to evolution, right? Therefore, increasing survival against selective pressures. And what are the selective pressures of a bacteria, everyone? It's not really food source and predators. What is it actually?
Antibiotics.
Yeah.
Antibiotics.
Very good. There you go. And if you mention all these points, if you mention it's not too important for pathogens, important for a host, the genetic material is DNA for a host, RNA for a pathogen, maybe you can mention that bacteria have prokaryotic DNA in their nucleus. Host DNA is usually enclosed in the nucleus in chromosomes. Apologies, bacteria don't have a nucleus. It's just a circular chromosome. And then you can mention host DNA, explain DNA replication, the life cycle there, and then link it to why it's important, because inaccurate replication gives you dysfunctional proteins, altered phenotype, disease, potentially cancer if cell cycle genes are affected, which can lead to death. Limiting, and this would be a really good thing to mention, you might remember continuity of a species is so important. So you can mention it limits continuity of the species. Whereas influenza, it uses glycoproteins and it uses host cell translation and host cell DNA replication. Talk about the enzymes. Then what is the effect? You mention that since they're not living, variation and mutation is important because it enables them to continue evading the host immune response as the antigens are always changing, and also evade pharmaceuticals, which are often enzyme inhibitors of viruses. And finally, for bacteria, you explain the bacterial life cycle. Very good that he mentioned binary fission. And then the effect.
Oh, Prithvi.
Yep.
The life cycles of pathogens and like influenza and stuff. What module is that in?
7.
Module 7.
Yeah, remember the first inquiry questions, all about the pathogens. What are the different kinds of pathogens and how do they cause disease? So if you want to revise that, go back to your Mod 7 notes to the different kinds of pathogens and you can look at it there. Good. Any other questions about this one? Okay. All right.
Because just that the first thing that came to my mind was like, you know how like they cloak themselves or something like that?
Yeah,
for
so
the
they can
body
have
to
an
not
envelope to evade the immune response. Yeah, but that's
so
an
that's the envelope.
adaptation. That's an adaptation. But that's not part of the
okay.
life cycle. Not all viruses are enveloped, right?
Mhm.
Yeah. So that's not essential to the life cycle, right? Out of all the people who can, be invisible, well, Harry Potter can with his invisibility cloak, but not all of us. And I guess it's a similar analogy with viruses. Not all viruses are enveloped. Only very few are enveloped. You don't have to talk about those when the question is broad and asking you about life cycle. Okay, good. All right, on that note, we have finished today's lesson. Does anyone have any questions? So all of you have your trials in how many days? 3.
Just less than a month for
13.
us.
Just less than a month
Yeah.
for us.
Oh yeah, bio. Yeah.
Yeah,
2
first-
weeks.
A month is crazy. That's too long because you know what's
But
going to happen
we
to all
start
of
our
you?
trials in 13 days.
Okay, yeah. So all of you start your
Like,
trials in
yeah.
13 days, which is
Our
the
bio exam is at like the very end but of
okay.
the block.
Okay, right, interesting. Okay. Who has a bio exam exactly in about 2 weeks? I think Serene, you mentioned yours is coming up very soon.
Yeah.
That's me too.
Okay, and Zachariah. Okay. If you finish sitting your bio exam and you have other exams to sit, just let me know. But I'd recommend probably not coming to class or watch the recordings. But you can spend that time doing extra study for your other subjects. Okay?
Okay.
Just so you can fine tune it. You're welcome to come, not kicking you out of the class, but I'm just saying it's probably the best plan for you if you've sat your bio exam in the following weeks to probably focus on your other subjects and watch the recording after trials. Okay? Because I'm telling you guys, trials, it becomes a bit stressful because some of you, does anyone have exams, 2 exams on one day?
Yeah.
Oh, that's a challenge.
Yeah.
I'm telling you, your hands start shaking. 6
Yeah,
hours
The
of
most writing ones as well. Like I'm doing the most writing in those 2 exams.
Yeah, so
English and SOR on the same day.
take it easy, guys. Take it easy and make sure you don't burn out. That's one of the other important things. When you come home, it's fine to take, an hour off, do whatever, and then before jumping into things. You don't have to turn into a productivity machine. Okay? All right. I'll see you all next lesson. Does anyone have any questions? Yeah.
So next week's lesson, are we just going to go through questions again?
Yeah, we're going to go through
yeah,
mod 8 focus questions now. So I'll
Mod
go through tech and disorders questions.
Yeah, so you want us to send in more questions if we have?
Yeah,
when we're going
keep
through
sending
that,
questions. Yeah, that's what we'll be doing for the next yeah. And then after that, I'll take you through any content points that you might have for tech and disorders. Okay.
What time is the
very
Saturday
quick question.
class?
Saturday class is 11 a.m. to 1 p.m. Saturday 11 a.m. to 1 p.m.
Right.
So since you've been added to the group, feel free to join that group and yeah.
Sweet.
You can
All right,
watch
thank
the
you.
recording, anything that works for you. Okay. All right, guys. Take care. All the best.
All right, thank you.
Thanks, Prithvi.
Thanks,
you.
Prithvi.
Thank you, Prithvi.
See you guys.